Hey guys, we're Proteomic Pathways, and we'd like to take this opportunity to tell you more about our Conrad concepts. The main goal of our proposal is to isolate cyanobacteria that can convert CO2 to biofuels, such as isobutanol. In doing so, we can address both global warming due to greenhouse gas emissions and the highly relevant global energy crisis. The first step of this proposal would be to have two communities of cyanobacteria. As you can see, one group will be grown in a CO2-rich environment, while the other will be grown in the absence of CO2. You may be wondering, what's the point of doing this? Well, extensive research has shown that when cyanobacteria are exposed to CO2, they express enzymes that convert the CO2 to biofuels, such as isobutanol, whereas cyanobacteria not exposed to CO2 do not express these enzymes. Thus, as enzymes are proteins, we can compare the protein profiles of the two groups of cyanobacteria to find the ones that are unique, which are the enzymes that can convert CO2 to butanol and its derivatives. What you're seeing right now is a protein profile. It's made by a process called 2D gel electrophoresis, which distributes proteins across a gel based on their mass and charge. After we've made protein profiles for both of the two groups of cyanobacteria, we can compare them with protein image analysis software, which will display proteins that are changed or different between the two protein profiles. Here's an animation showing how exactly enzymes work in converting CO2 to isobutanol. Basically, each enzyme is responsible for a specific step in the conversion, and they collectively convert the CO2 step by step into isobutanol with intermediate compounds along the way. A big part of our Conrad concept is identifying these enzymes. Once we've identified these enzymes, though, what use would they have? That's the next step, identifying the specific species of cyanobacteria these enzymes came from. Here's an enzyme. Since proteins have unique amino acid sequences, and amino acids come from DNA, proteins also have unique corresponding DNA sequences. We can synthesize complementary strands of DNA that would bind to cyanobacteria that contain a protein of interest, since proteins, as stated, have unique corresponding DNA sequences. These strands of DNA, shown in red, would have fluorescent molecules attached to them. After these fluorescent DNA probes are injected into the CO2 group of cyanobacteria and bind to the cyanobacteria that our protein of interest came from, they will fluoresce under UV light, allowing us to easily isolate and identify the bacteria they came from. Once we've identified these bacteria, the next step would be to test them for efficiency and seek industrial partnership to integrate them into biotechnological farms for massive biofuel production. We believe that proteomic pathways can provide the green and powerful energy we need to sustain our future.